Good morning, girls. How are you all?
Good morning, Miss.
Good morning. You have come on time. I have
Please.
seen few girls. Now it is 5. It was 6. Anyway, no problem. It's just 12 o'clock. No problem. Today, what we are going to do, see, first we will solve 62 May, June 17. And then that February, March, the variant, the few pages, that 17, the February, March 17 variant, few pages we didn't do, that variant we have to solve. So girls, that I'll share the screen. Look at the screen. This is the 62 variant. I don't know that girls are taking time to join. Okay, now let's start. The first question, you need to read the first question. Everybody look at the screen. I'm showing the past paper. First, if you look here, a student wanted to investigate the effect of different growing condition on the rate of photosynthesis in plants. When photosynthesis takes place in a leaf, oxygen gas is produced and it released into the air spaces in the leaf. Carbon dioxide will combine with the water and it will produce glucose and oxygen. When small discs are cut from the leaf, small pieces, okay, the gases in the air spaces in the leaf disc can be removed. This allows the leaf disc to sink into the places they are placed in the liquid. As photosynthesis takes place, the leaf disc start to float. Why? The time taken for the leaf disc to float indicates the rate of photosynthesis, because it will in this process they are going to produce this. The student selected two plants, X and Y, where were the same species, but their growing conditions are different. Two different locations. They have taken the same species leaf. One is X, one is Y. One leaf were removed from plant X, and 5 small discs were cut from each of them. And each leaf disc was 8 millimeter in the diameter. Now the student used that they have placed the 5 discs from the X into a syringe which containing 5 cubic centimeter sodium hydrogen carbonate solution, and another one into another syringe. The student removed the air from the air spaces. Look at the syringe, the way they are removing the air spaces into the leaf disc from plant X by pulling and releasing the syringe, the plunger, the way it's shown in figure 1.1a. The leaf disc then sank to the bottom of the syringe as shown in 1.1b. Look, they have just removed the nozzle part that keep with the finger, and slowly they'll pull the syringe to remove all the air. Now the leaf is look at this, that leaf, it is sinking in the syringe. Now second page, the 5 less discs, the pieces, small pieces from the plant X were removed from the syringe, and each one has placed into separate test tube in a test tube rack. 2% sodium hydrogen carbonate solution was poured into each of the test tube to a height approximately 5 centimeter in the bottom of the test tube. Look at the picture. From the bottom, 5 till 5, and 2% sodium hydrogen carbonate they have that kept. But instead of measuring the height, measuring the volume of sodium hydrogen carbonate will be more accurate. And by looking this diagram, you can understand that, look at this picture. Here, the 5 test tubes, that a lamp was placed at a distance of 10 centimeter from the edge of the test tube rack. This is showing figure 1.3. So don't you feel, girls, that from the lamp, the temperature, the test tube 5 is getting more than the test tube
1,
right?
Test tube 1 is getting less. Yes, Miss.
Right, girls? Now,
yes.
So this is the reason.
It just looks like solar system. Mercury
this
being
is actually error.
Yes.
This is one type of error. Now, step 7. The lamp was switched on and the timer was started. The time taken for each of the 5 lift discs to start to rise to the surface of the liquid in the test tube was recorded. Now, the student repeated this whole process from the lift disc that they have taken from the species, the lift Y. You can understand what they have done. Now, this is the plant lift disc X and the plant lift disc Y. They are asking you to prepare a table now I will make this table in the whiteboard. So for this thing I need to share. I need to stop sharing. I will show you how to do this with whiteboard. I need to draw the table first. You can do by yourself also, no problem. It's not becoming straight, no problem. This is lift disc 1, 2, 3, 4, 5, 5. Total 5 maybe, right?
Yes, miss.
Total 5. So I need to use eraser just to remove this. So I will make it 1. So as my drawing, just don't think about my drawing. So here, after that, here will be plant X. My tables are not uniform. You just make it with the scale because whiteboard is not good to write. Now here will be time slash second. Our unit will be second. And here it will be plant. Oh, but I couldn't make it properly. Again, this test. Now, again, this text here will be
Disc.
That this is X. Here will be Y. Now, X and Y, and here lift disc. And if you are writing it, lift disc 1. Another line you should make.
And lift disc.
Yeah, DISC. Disc 1. Okay. Disc 3.
4, 5.
Disc 4. And the last is disc 5. I need to extend it. Disc 5. So this is now what I need to do. I need to draw one more line here. So here will be one more line. What happened? Now you can understand this 1, this 2, 3, 4, and 5. Now you need to calculate the timing. And hey, who is drawing?
Miss, I was asked about
plant X. Disc 1 is 1 minute 10 seconds, so it will be 70, right? And then disc 2 for plant X, it was 1 minute 5 seconds, so 65. And then the disc 3, it is 58 second, and then disk 4, it is that 1 minute 26 seconds, so 86, and then disk 5. Disk 5 will be 1 minute 18 seconds, so 78. Now see, you have done it. Now again, the text, and I'll start from here. So plant Y, girls, plant Y, that first one is 3 minutes 5 seconds, so 185. 185, and then disk 2, it will be 252, and then disk 3, it will be that 4 minutes 10 seconds, so 250. Now then disk 4, it is 4 minutes only, so 240, and then disk that 5, it is 3 minutes 37 seconds, so 270. Oh, what is this? 270. Okay, girls, is it clear? The table, yes, you understood?
Yes, miss.
Yes, Neha, good morning. So this is the table that you have prepared in this that in this space they have given in page number 4. Prepare a table to record the results showing in figure 1.4. Record the students' result in your table. So I hope you have done. So I will stop sharing this and go back to the question. The question we were, I'm going to share you again. So we did this table. Now we will going down. Anika, the answer you have given to me, I checked it is correct. Now
correct,
girls,
miss?
yes, correct. Now girls, this one, this I want the answer from you. Calculate the average time taken for the lift disc from plant X and the lift disc from plant
All
Y.
the time divided by 5.
Yes.
Although,
so for the plant, you need to provide your answer as a whole number. Okay. You need to use it as a whole number, and I want you people will give me the answer, please. 70 plus 65 plus 58 plus 86 plus 78 equal to 357 divided by 5. Do it and tell me the answer. Average answer for X.
miss, is it 71.1?
Excellent. And the answer whole number will be 71 seconds. Okay. And what will be plant Y? Yes, good. Someone is answering me, Zareen. Thank you, Zareen. 71, 229 seconds, 71 seconds. Excellent. But you have to show the working here. Okay, thank you, Zareen. Now, after this, there is a graph. In this graph question, graphical representation, you will tell me first which one is X, which one is Y. Girls,
Is plant X is
and
X.
plant Y will be X, and the Y will be average rising time / second. So I will give you this that answer. I'll show you the answer. You people try in between. Have you seen, girls, the graph? Have you seen?
Yes.
The graph plant X and Y. So the average rising times / second. So do you have the paper in front of you? Shall I give you 5 minutes to draw the diagram?
Miss.
Yes, yes, do it. Okay, done. And here, one thing, girls. Do you think that I have used here 2/3 of the graph? I used 2/3, but you can extend it a bit more. Okay, by instead of giving the gap 5, you can give 10. Then your graph will be more extended. Done, girls?
Miss, do we send it to you after doing?
No, if it is correct, no need, because but I'm just telling you from plant X and plant Y, you can extend your graph. Starting time, I have given you here the 5 square gap. You can give it 10 square gap. So again 10 that is square for the graph for plant X and 10 square gap. Then your graph will be more extended. You are using the area will be more.
Done, Miss.
Done. Okay, so easy. Bar chart, this graph is very easy. Thank you. Now, I will come back to the question so we have finished the graph, and next question, see what we are going to do. And next question, they're asking, suggest why the lip disk rise when the photosynthesis takes place? Because
the gas expansion.
It produces oxygen, right, during photosynthesis, which this oxygen trapped in the lip surface and reduces density, increases buoyancy. Yes. One mark, no need to write so many things. Just write that it reproduces the oxygen, which reduces density and increase buoyancy. That's why it's float. 1 point is enough. But oxygen, it produces oxygen, which is trapped in the lip space and reduce density. This much you have to write. Now, explain why 5 lip discs of each lip type were used.
So that the results will be more accurate.
If it is 2 marks, then you will write like that to get the accurate result, accurate average result, and to identify anomalies. But 1 mark, 1 point is enough. Now identify the variable that was measured. What did you do here? In this experiment, what are you measuring?
Light.
You are measuring
Light
the time.
intensity.
No, you are measuring here the time taken for the lip to rise.
Oh, okay.
Right? Or
Yes.
And what are the things you have taken? You have taken the lip X and Y from different location, right?
Yes.
So the measured thing, if you're writing rate of photosynthesis, also correct. If you're writing time taken for the leaves to rise, also correct. And change thing means location of the plant or the growing condition of the plant. And if you don't understand this, we will go back to the question, first page. And look here. Step 1 is clearly written which were the same species but were growing to different location. So the change thing is, you understood that the things, that's why you need to read paper 6. The first 2 pages are very important. All the information in the next pages has been given in this first 2 pages. You need to read carefully. So the measured, if you don't understand rate of photosynthesis, if you're writing time taken for the leaf to rise, also correct. And the change, growing condition of the plant or location of the plant, both are correct. Now, state 2 variable that were kept constant. It means in this
Volume of water and size of discs.
Size of the leaf. Size of the lip disc, okay, and the that
volume
concentration. That water they have used. Did they measure? Look at this. They have used 2% sodium hydrogen carbonate.
They use same volume of water, Miss, 5 centimeters. See.
5 cubic centimeter, yes. No, that this was not water. This was 2% sodium hydrogen carbonate solution. Which
okay,
is
so volume
clear.
of solution.
Volume of sodium hydrogen carbonate, or you can write concentration. Okay, distance of the lamp also you can write 10 centimeter, right? But that is an error. If you're writing because they were kept constant, look at that is step 6, 10 centimeters. So distance from the lamp or the light intensity also will not be wrong. There are
light intensity will be wrong, Miss, because 10 centimeters is from 5th rack, not from 4th one.
Listen, that is the error.
Yes, miss.
That we will explain in error. But if you are writing light intensity or distance of the lamp will be correct in this point, because they're asking what was the variable that was kept constant. In the test tube 5 or the test tube 1, that is error. That we'll explain in the error time. But now the distance is fixed. We are not going to tell that it is fixed for test tube 5, not for 1. No, we will tell light intensity due to the fixed distance from the lamp. That is because they showed you, they told you the distance of the, if the distance fixed means light intensity also will be the point. If you're writing, you will get marks, but error will be there. That's true. That's why when they're asking this, if you're going to the next question, error. First error is, see that the lift disc are different distances from the lamp. Actually, whenever you will not get the point, you have to use this point, right? So error in that time, the point you are asking that light intensity is wrong. In this point, you will write it. Okay, but they have kept constant, but maybe the way they have kept constant is not correct. So in this point, we will kept in this error. How you are going to write it? You will write lift disc are different distances from the lamp. And improvement, arrange equal distance or do the test for each test tube separately. Okay, so and one more thing, see, did you notice that they have measured the height? I told you that time, right? Look at this point. Look. 2 cubic centimeter hydrogen carbonate solution was poured into each test tube to a height of approximately 5 centimeter. I told you in that time, measuring height always is an error. It is not 100% correct. So we will be writing like that, that measuring height, not the volume of sodium hydrogen carbonate. This is the error. And the improvement, measure the volume of sodium hydrogen carbonate by using a measuring cylinder.
Okay.
Okay. Look, I'm telling you the error again. Lift disc are different distances from the lamp. And improvement, arrange equal distance or do each test tube separately. And another error is measuring height, that's not the volume of the hydrogen carbonate. So measuring the volume by using a burette or measuring cylinder, whatever you feel. We have completed 19 marks, that question number 1. Now look at this question. This is that 42 micrograph or the cross section of a leaf taken from different areas of the same tree. Some parts of the tree are shaded and some parts are getting the sunlight properly. So sun leaf one shaded leaf. So you can understand the shaded leaf and sun leaf, their adaptation, their arrangement also it will affect. The cross section shown in figure 2.1 was taken from the leaf grown on the shed. And the second one, the cross section, this one is direct sunlight. Look, leaf grown in direct sunlight. And this one is leaf grown in the shed. So actually, you know that sunlight is less photosynthesis, less food. So their leaf structure also shows some differences. Now, 2 visible differences. Girls, what are you seeing?
Spongy mesosphere.
Look, this 2. One, this one, and another one is this. Many you can write. See, sun leaf palisade cells. Look at the sun leaf palisade cells are taller and thinner than that shed leaf, right? You can write the leaf grown in direct sunlight has bigger cells, larger air spaces. Look, the leaf grows in the direct sunlight. It has larger air spaces than the leaf grows in the shed. And then you can write the sun leaf palisade cells are taller, thinner, or also you can write palisade mesophyll layer. Spongy mesophyll layer is thicker in sun leaf. Look, any 2 points you can write from this 2 diagram, but make sure you are writing what you are seeing. Don't write the things which is not clear in the diagram. Look at these vascular bundles are also here. Now, girls, next question they're asking, make a large drawing of the leaf cross section shown in figure 2.2. Do not draw individual cells. Do not label your drawing. Thus, this question came in 1962 variant, and my students were telling, Miss, unseen diagram we didn't do, but it is same diagram. Problem is diagram you are not practicing. This is the problem. And they're asking, Miss, if they're asking do not draw individual cells, then what will you draw?
Miss,
Actually,
did they say unseen?
Yes, they told me unseen.
Why, Miss, is just in front of your eyes. What is this?
So that those who are right now that grade 11, few students told me, Miss, that the diagram was very hard. It's very hard. That's true. Actually, it's not very hard. It is time consuming. So you need to practice within 10 minutes. You have to complete this diagram. You will not get more than 10 minutes because you have to finish your whole question. Now, so here, what does it mean? Do not draw individual cells. Means you will not draw 1, 2 cells. You will draw the bundle of the cells packed one after one like that. The way they drew, so that you can make it clear, the layers. Understood? So when you draw the upper epidermis, don't draw one cell. And all the cells will be stick to each other. And you will draw lots of like that palisade mesophyll cells so that it will be clear for the examiner that this is the layer of the mesophyll. Then go to the loosen, that spongy mesophyll area, but you have to make sure it is connected. No, not a single cell will be individual. All will be connected with the layers. This is the answer. You just try at home and you can send this diagram picture if you want. And that thickness, this is related with the diagram. If you don't do the diagram, you cannot. Length of PQ, girls, if the diagram is not in front of you, you cannot. Length of PQ, but I have measured it 19 millimeter. And actual length 19, and then what is the magnification? Magnification is 130, right? 130. So 19 by 130. 19 divided by 130 is 0.15 millimeters. So 2 decimal place they told. So I gave you like that. So this is this diagram. Now, this page is also you can do by yourself. Look at this question number B. A student measured the length and the thickness of some leaves taken from different tree. Some parts of the tree were direct sunlight and some parts of the tree were shaded from the sunlight. This is the diagram: leaf grown direct sunlight, leaf grown in the shaded side. And shaded side leaf will be taller and thinner but because they are thinking that if they grow faster, they will get more sunlight. The severe condition, we call it titillation. You know this. Now, they're asking the average length. And girls, this is average length millimeter. This is, this is actually, you have to subtract 105 minus 70. What is the differences? The difference will be 35, right? And the percentage differences, can you remember that formula? I'll show you in the whiteboard. You just check this.
New value by old value.
Yes, that's the formula. Now I will show you here in the text box. I'll show you here 105 minus 70. Okay, who is drawing this all? Shall I remove you? So what is this? This will be 35. 35. This 35 you have to divide by 70. Same formula, that percentage formula. And you will get 50. So this is the result of this. Calculate the percentage difference between the average leaf length of the leaf grown in direct sunlight and the average leaf length of the leaves grown in shade. So the average, that the percent differences, one they have done for us and one we have to do. So for this work, you are going to get 2 marks. Now, where was our question? So you understood this, the space for working, what you need to write. Now, the next question is, the student was testing a hypothesis and stated, the leaves grown in the shade will be larger than leaves grown in direct sunlight. Now, you need to give some evidence which support this hypothesis, and you will need to give some evidence which do not support this hypothesis. It means positive and negative, right? So leaves grown in shade will be larger than leaf grown in direct sunlight. Let's look at the leaves. Positive thing, if I'm telling, can you look at the diagram? Leaves grown in direct sunlight is shorter and the shaded leaves are longer. So that sun leaf is 18 millimeter length and the Shaded leaf is 35 millimeter length. So can I say the leaf grown in shaded side will be larger than the leaf grown in the direct sunlight? Supported
Yes.
the hypothesis, right? Second one they're asking does not support the hypothesis. Leaf grown in the shaded side will be larger than leaf grown in the sunlight. So if we want to say doesn't support the hypothesis, look at the leaves. Leaves grown in sun are thicker and wider than the leaves grown in the shade.
Yes, miss.
Right? So this is the way you have to explain the answer of this question, leaves grown. Now, next page. This, before testing the leaves for the presence of the starch, the green chlorophyll must be removed. The chlorophyll can be removed by boiling the leaf in ethanol. Describe how ethanol can be boiled safely. What you're going to do, you will place this ethanol into a test tube and boil the whole test tube and heat it in the water bath. Do not heat into the burner or direct the flame, okay, because it is flammable, right? Alcohol is flammable, that's why. So you will heat it inside the water bath. So place the ethanol in a test tube and boil it in the or heat it in the water bath. Don't heat it directly with the flame. And next one, suggest why chlorophyll need to be removed.
Because it can give anomalous result when you put the iodine solution there.
Actually, you will not be able to see the color change.
Exactly.
It's not anomalous result. You will not be understand actually. Due to the presence of chlorophyll, when you are doing the starch test, it will give you the blue-black color, right? In presence of this chlorophyll, you will not understand.
See what color is coming.
Color change. Now, girls, here is a planning question. Can you remember the plant nutrition chapter, grade 8? We did it, right? And here we did the starch test also. The answer I have given over there, this is the same answer, only the first 2 line difference. A student wanted to find out if the starch was present in both leaves grown in direct sunlight and the leaves grown in shade. Plan an investigation to determine if the starch is present in both the types of leaves. So of course you have to take the D-starch leaves, right? So the first 2 line, others, everything exactly grade 8 notes. See, take the leaves from same plant of the same species. Take at least 2 leaves from sun and 2 leaves from the shaded part. Now drop the shaded and that sun leaves separately into the boiling water into different water bath for 30 minutes. So the enzyme will denature and it will prevent further chemical changes. Remove the leaf and drop them into a tube of alcohol or ethanol and heat them. Hold the test tube carefully with the tongue and heat it in the water bath. And heat until all the chlorophyll has come out from the leaf. Then remove the leaf from the alcohol, dip it into the hot water to make it soft, because after boiling in the alcohol, it will be brittle. Then take a white tile, spread the leaf and add iodine solution. The blue-black color shows the presence of starch. Use same volume of iodine solution and heat them in that same time to get that great result. See, girls, the whole answer you have in grade 8 plant nutrition chapter, right? Only the first 2 lines, because they're asking to determine if the starch is present both types of leaf. So you are taking 2 different take leaves from same plant or species, at least 2 leaves from sun part or 2 leaves from the shaded part of the plant. Because they are asking both leaves grown in direct sunlight, leaf grown in shade. So you're taking 2 leaves which grow on the same species, same plant, but 2 leaves from shaded part, 2 leaves from the direct sunlight part. After that, carry out the starch test normally. Thus, this is not food starch test. This is the leaf starch test. So the whole steps you need to give. What are the steps? First, you will boil with the water. So enzyme will denature and it will prevent the further chemical changes. Then you will take out after 30 minutes to remove these leaves and you will dip it into the alcohol in a test tube and hold the test tube carefully with the tongue and heat this in the water bath.
So the starch will be present in the leaf which was in shade, right?
Yes. And that's the already practical, thus no need to tell the answer directly.
Oh, we won't tell that which.
You will tell that part, the blue-black color shows the presence of star. Use the same amount of iodine solution and same time of heat, heat them in the same time so that
But means the whole thing is same, just like the copybook. What's different?
Yes, only that first two line, and the last line you will use the same same polymer iodine solution, and that
First two line
use
was
same
same
time.
as well.
Fine,
It's
first
just
two
about
line and the last two line will be different because it according to the question. This is for every question. Even though the same question from the copybook is coming, whenever you are writing the answer, you have to blend the question and the answer. So starting 2-3 lines will be different than the same from the copybook.
okay.
Okay, girls, I hope this variant, see, we have completed this whole variant.
Wow, before time even.
Yeah, so I don't
Half an
think
hour.
so is the problem. I have less than 1 minute, and you people start this 17 February March variant. After this, I will do that. Okay, girls, I'm coming back.
Oh, miss.
কি হয়েছে?
Who has a baby sister or something? Nihat. আচ্ছা।
Yes,
কেমন আছো?
বালা।
Miss, we were in question number 5. This question, Miss. Miss, your microphone's off. Yes, it is. Everybody's microphone's off. It's just me talking. Wow, weird.
Okay, thank you. Now, everyone, can you hear? Girls, are you
Yes.
listening? Okay. Now, I don't know how it becomes mute. Look, the organ that breaks down alcohol in human body. What is the name of the organ? Liver, right? Now,
Yes,
if we are going down, look at the 3D shape of this enzyme. These also most probably I have discussed with you on that day, but I'm not sure. That's what I'm telling you again. Explain why the shape of enzyme is very important. Otherwise, because enzyme has a specific shape, enzyme must, the active side of the enzyme must be complementary, the shape should be complementary with the substrate. Otherwise, they will not fit together, they will not form enzyme substrate complex. And if they're not forming enzyme substrate complex due to the different active site, they will not work out. Enzyme will not break down the substrates or not build up that substrate. So it will not work. That's why the shape of the enzymes are very important. This question also from the notes that explain why an increasing temperature causes an increase in enzyme activity. Increasing temperature increases the kinetic energy. So the molecules move faster. It also increases the frequency of the collision and increases the number of successful collisions. So enzyme work faster. Girls, this is this notes also you'll be getting in enzyme chapter. Now, state one factor other than temperature, pH, right? That also affect the enzyme activity.
Yes.
Define that. Yes, miss. You need this
No,
answer?
I said yes, Miss. I agree. No, Miss. I agreed with you.
Okay, thank you. Now, define the term gene. That is the, the common question
Length
from
of
this
DNA.
coordination chapter. Length of DNA that codes for a protein. Now, describe the role of ribosome in the synthesis of proteins such as enzyme. You know this thing, mRNA copied the messages from the DNA and passes through the ribosome. Ribosome read out the messages and assemble the amino acid into the protein. This is also from that coordination chapter. Now, look at this. Question number 6. Could you please tell me the name of the enzyme which break down the starch and
Amylase.
what is the product? But remember, your gland is salivary gland. So you cannot write here sucrase, lactase, like that enzyme name. So
That's obvious.
Saliva contain 3 things, right? One is
Yes.
water, one is mucus, one is amylase enzyme.
Amylase.
Okay, so what the function of water? It dissolves the food. Mucus makes it slippery, help to bind the food into a bolus and makes it easy to swallow. So enzyme is amylase. Starch will turn into what is the product?
Saliva. Product is maltose in starch.
Excellent. And thank you that you didn't tell me the glucose, because some students are immediately they're telling glucose, but glucose, it will take time, girls. So starch will turn into maltose. Now, maltase enzyme. Maltose.
Substrate.
What is the product?
Product is glucose.
Excellent. And location of this maltose, where it's
Small
separated? Small intestine wall. Excellent. This maltase will secrete in a small intestine wall. It will not secrete in the stomach. Okay. Now, amino acid in the stomach wall. So, what is the enzyme? It means that protease enzyme, right? If the product is amino acid, pepsin. Excellent. And the substrate will be protein. Protein break down amino acid, and it is forming from the stomach wall. Now, again, pancreas and small intestine wall. Another enzyme, that is trypsin. And the substrate is protein, amino acid, will break down amino acid. Actually, in that time, sometime many protein couldn't finish the breaking down process. They become that peptide. And peptidase enzyme break down it finally. Now, lipase enzyme, substrate is fat.
So what will be glycerol.
And where it is, the location? Pancreas. You can write a small intestine, you can write pancreas. Now, girls, bile made in liver, stored in gallbladder, and passes to the small intestine. You know this, the role of bile in digestion. See, this is complete chemical digestion. This is not a chemical digestion. You need to write like that. Bile is a yellowish green substance which produced in the liver and stored in the gallbladder. Through the bile duct, it comes to the duodenum. It neutralizes the acidity of the food molecules and also break down the large drop of fat into small droplets, which is known as emulsification. It do not contain any enzyme, so it do not change the food molecule chemically. It just break down into small pieces and increases its surface area so that the lipase enzyme can work well, and the fat digestion could be faster. This is the role of bile. Clear, girls? I told you so many things, but you just need to write the answer properly. Now, what is absorption?
Movement of small food molecules into the blood.
Through the wall of the intestine, right? A small intestine into the blood, or into the lymph also it is going, right? Through the lacteals.
Yes.
See, it is not even 1 o'clock. Look at in our area, the police car, the way they're, see, you are hearing the siren sound?
Yes, miss. Yes.
It's really, that is so scary that you couldn't be able to open your windows. Sometimes I feel like that. What happened, today morning, I gave that food through the window to the cat. And that cleaning people came and they cleaned that. So cat couldn't get the chance to eat the food. So I'm keeping the food in an open way, just throw the slowly from the window. So it will fall on the ground, because now car, everything is fixed. The space was empty. So that man who came to clean that area, he cleaned that cat food also. Now I'm planning that I will give after the that 2 or 3:30, but I'm scared to give that also. If they're looking that we are opening the window and seeing, it's like lockdown, right? It's complete lockdown.
Live in a very busy area.
Someone is asking my leg. Yes, I'm fine. I can walk well, but I couldn't get down through the stairs. Still, I'm getting pain if I'm only going down through the stairs. Others, everything is okay. Yeah. Walking is not a problem, but getting down through the stairs, still, I couldn't getting down properly. I need to do one by one, slowly.
Sprint takes time, literally.
Yeah, it will take time. Yeah. And you know that my age, that you people are small children, your healing process and my healing process is not same, right?
My age is not working. My age is even not working, to be honest.
No, age is a factor. Age is a factor. Healing process will be faster in your age. And as my body mass is more, it put more weight on my feet, right? So that is also one matter. Second thing, and is the healing process also due to the age. It's not like your people, right? And thank God I don't have the sugar problem and all. If any person is having sugar problem, it could be double.
Why are you like age and stuff? You're still young.
I feel like that. Thank you. Okay, next question. Lack of protein in diet can result in protein and energy, one type of malnutrition problem. What is the name of this? Marasmus. Today, I showed a marasmus child picture to grade 8 girls. Okay, and see that I'll show you today, you also, just a minute, I'll show you this diagram, the marasmus child. So this was in, are you, girls, is it clear, the diagram? All of you are seeing this diagram, marasmus child?
No, miss, we're seeing your page.
Okay.
You can stop sharing and then share again. Then we'll see.
Okay. Look, I will share again. Look this. Now it's clear, right? Look at this
Yes,
child,
miss.
marasmus
Allah.
child. This child is that below 10 years, 5-6 years old. I don't know from which country. They are telling after the war, it was severe shortage of food. And this child, look at, only the bone is covered with the skin only, no flesh. So this boy is not be able to stand even, look. So the severe lack of that energy causes this kind of problem. This type of disease, in your question also it's there, lack of protein in the diet can result protein energy malnutrition. State the name of one disease caused by protein energy malnutrition. If you're writing kwashiorkor, you know that pot belly, right? The belly becomes swell, but the child, it looks very thin.
Emaciated.
The hair color also change. And the marasmus child, this is the picture of the marasmus child. Now come back to our question again. So this question, we already solved the last question. See, people that eat they eat a diet that is high in fat often advised to reduce their intake of fat. Suggest the health benefit to change their diet. See, this is a simple question, right?
Not be obese or get
simple
CH.
question. Yes. So if you are changing your diet, see, nowadays is still now all the doctors are telling that as there is no vaccine for this coronavirus, immunity is the only thing that we could rely on it. So immunity, do you think that within a night, and nowadays what happens if you are going to the panda, no orange, no lemon, no ginger? Okay, so people are buying even the ajwa dates also will not get. So what are you thinking, that taking this all within 1 day, 2 days will increase your immunity? No. This is a series of practice. If over time, if you have a practice of taking healthy food every time, you are taking fruits, vegetables, you are taking less amount of fat, you are doing half an hour exercise, minimum 5 days a week, okay, your metabolism and your body weight is perfect. These all boost up your immunity. Within that 2-3 days or within that 1 week, 2 weeks, if you are thinking that eating healthy food will boost up your immunity, you will feel good, that's true. But it will take time. So that's why in my class, I always, that told all of you that don't take outside food or reduce the amount of this outside food. Take the food, the fresh fruits and vegetables. Make sure that your food contain minimum 5 types of different fruits and vegetables. And the fruits and vegetables have different types of color. It's very good for your health. And if you're taking it regularly, your immune system will be good and you will not suffer with this type of contagious problem. You will less suffer. Even though if you get any kind of disease, your immune system will protect you. So if you do care yourself, that your immune system will also care for yourself. Many people are having very unhealthy lifestyle. They are taking this fast food, like all types of, that roasted chicken, burger, French fries, cold drinks frequently. There is no limit of taking this kind of food. So, okay, once in a month, twice in a month is okay, but every time is not good, right? And at least you people are a small age, so nature is protecting you. But in our age, when the people will be 30 plus, we should stop this all, definitely. Because nowadays, nature is not protecting us, right? So young people will get some benefits due to their age, but those who are 30 plus, they should maintain this healthy habit. Now, what kind of health benefit if that person will get if they have changes their intake of the fat? See, it will reduce the calorie or energy intake, right? And it will reduce the body weight and the obesity problem also that person will be, it will reduce. And also the important thing, it will also reduce the chances of coronary heart disease or CHD, to get it right. Even you can write the stress on the joint will be reduced, cholesterol level will be reduced, okay. And if many obese people are having breathing problem, organs are working more properly, hormone level will be normal. These many
They get
functions.
tired easily.
Yeah, that you can do more work, that if your weight is idle according to your height and age, you can do work more than the obese people. So guys, this is our that 17 February March. I hope that you have understand the last part of this paper 4 and full one variant of paper 6 I have completed. Next class, I will do paper 6/3 variant, because 6/1 variant I did. I have checked. 6/3, May, June 17, I will give you the PDF question.
Okay, miss.
Okay? Any
Miss?
question? Yes, miss?
Oh, this is not related to this, but what's our optimum weight, like for our age group? What would be the
It's
optimum
that,
weight?
listen, you have to BMI, calculations are there. You have to go to malnutrition, animal nutrition chapter. Your height, you'll convert it into meter. There is a formula. You can check the Google, you will get the formula. You can, if you divide, that 5 kg here and there is still doctors are accepting, but it is a problem.
Miss, what's the formula again?
Formula, do the Google. That is, I need to check the textbook with that obesity, that BMI, basal, that metabolism.
So if I write in Google, I'm gonna get this?
Yes, there is a formula. Your height, you have to convert it with the meter, and then square divide with the weight. There is a range that below that 23 is clean, to like up 24 to 30 is maybe normal, 30 plus. Check the Google. You can get, there is a range of this answer also. So girls, this is our today's that class, and next class I will give you the link before the class. Okay, and you have 3 classes every week, starting from Tuesday, Wednesday, Thursday. 3 classes a week. Don't take the stress of the exam. Just continue your studies. Let's see what happened on the 7th April. Hope you all will get a proper justice, inshallah.
Inshallah, miss.
Okay, girls. Thank you.
Thank you, miss.
And bye, everyone.
Have a nice
weekend.
You too,
I don't
miss.
think so there is any more nice weekend. We can't go out.
Staying home, huh?
And